Hello everyone, welcome back to the class. In our previous class we discussed about the metaphase stage of mitosis cell division. As we know, metaphase is an extremely dynamic part of the cell cycle where the spindle fibers rapidly assemble and disassemble as they grow out of the Mitosis, seeking out attachment sites at chromosomes. To attach to spindle fibers, each chromosome has a specialized structure called kinetochore. So it is located at the centromere. There are two kinetochores on each chromosome, one on each sister chromatids facing in opposite direction. So how does the kinetochore become attached to the spindle fibers? So it is through a process called as search and capture, in which the plus end of some of the microtubules radiate outward from the MTOC and attach to kinetochore. Some microtubules randomly hit kinetochore and are subsequently stabilized to form kinetochore microtubules. So eventually the opposite kinetochore on the other sister chromatid will be captured by the free end of the microtubule from the opposite pole. So this leads to bipolar orientation of chromosome. Okay, and kinetochore microtubules remain in a state of dynamic flux. The plus ends of kinetochore microtubules can add and lose tubulin subunits while attached to the kinetochore. So there is a specialized protein. The protein is microtubule dependent motor protein. So the role of microtubule dependent motor protein in metaphase is that the movement of chromosomes during metaphase depend on numerous microtubule dependent motor proteins. So these proteins belong to two families, that is the kinesin related proteins such as kinesin 5, kinesin 14, kinesin 4 and kinesin 10, and the other family of protein is dynein. Kinesin 5 protein is a plus end directed motor protein which interacts with the plus ends of antiparallel polar microtubule in the spindle midzone and push the poles apart. Kinesin 14 proteins are minus end directed motor proteins in addition to their motor domain. So they contain other domains that can interact with another microtubule, enabling them to cross-link antiparallel polar microtubule, the spindle midzone, and slide them past each other and tend to pull the poles together. While kinesin 4 and kinesin 10 proteins are also called as chromokinesin, are the plus end directed motor proteins that associate with chromosome arms and that push the attached to chromosome away from the pole. So dynein is the minus end directed motor which links the plus ends of astral microtubule to components of the actin cytoskeleton at the cell cortex. The dynein motor pulls the spindle poles towards the cell cortex and away from each other. And we also discussed the different forces which move chromosomes during metaphase. So at metaphase stage, chromosomes aligned at the midpoint, that is equator between the two spindle poles. So during this process, chromosomes often oscillate backward and forward before arriving at the midpoint. So there are multiple forces which move chromosomes at the equator. One major force is the force which is associated with the kinetochore. So the kinetochore and its associated chromosome along with the kinetochore microtubule toward the spindle pole will produce this force. So it is produced by proteins at the kinetochore itself. So depolymerization at the plus end of the microtubule somehow generates such force that pulls the kinetochore poleward. Kinetochore localized kinesin 13, which is a class of microtubule depolymerizing protein, and CENPE, which is a member of kinesin 7 family, plays an important role. And apart from this poleward force, so there is polar ejection force, which is the second type of force. The function of this force is it pulls the kinetochore. Just a minute. It is a force which acts on the chromosome. So Each chromosome is simultaneously subjected to anti-poleward force. The force acts on the chromosome, okay, plus end-directed motor proteins such as kinesin 4 and kinesin 10, which are located on the chromosome arms, are believed to interact with the polar or astral microtubules to produce the anti-poleward polar ejection force. So this force pushes the chromosomes towards the equator so that the metaphase, the chromosome will be aligned on the metaphase plate. So this alignment of chromosomes at the metaphase plate, midpoint between the two spindle poles, is commonly called as congregation. So this was about the metaphase stage of mitosis cell division. So in today's class, we will be discussing about anaphase stage of mitosis cell division. So anaphase stage is the shortest phase of mitosis. So as mentioned, each sister chromatids of the metaphase chromosome is attached to the kinetochore microtubules via its kinetochore, right? The opposite ends of these kinetochore microtubules associate with one of the spindle poles. It may be left spindle pole or it may be right spindle pole. At anaphase, the kinetochore microtubules pull the two kinetochores towards the opposite poles, okay? See, each sister chromatid of the metaphase chromosomes is attached to the kinetochore. So to draw, so there is chromosome attached or aligned at the metaphase plate, and there is kinetochore which is present at the centromere at the close proximity, and there is a spindle pole here and spindle pole here. It is helping the microtubules to protrude out. Plus ends are towards the center, away from the pole, right? So this microtubule gets attached to what? Gets attached to the kinetochore, or simply I can draw like this, gets attached to the kinetochore. So this becomes kinetochore microtubule, to which the kinetochore is attached to the microtubule, right? So this will pull the associated chromosome to opposite sides, then sister chromatids will divide, or sister chromatids will separate. So this process happens in anaphase stage. So there are many dynamics happening here. So sister chromatids starting. What are sister chromatids? To be clear, so this is the whole chromosome. Sister chromatids are these. So this is chromosome first. I'll write it again. So this is chromosome having 4 arms, 4 chromosomal arms. So this is chromosome. When this chromosome becomes into sister chromatids, so sister chromatids is this. This is sister chromatids. Sister chromatids. So this is done, or this is separated. Chromosome is separated into sister chromatids by anaphase. Okay. So these sister chromatids do not separate. So they are held together at their centromere by a multi-protein complex, which is known as, you know already, that is cohesin. So cohesin is the multi-protein complex, a protein which holds the sister chromatids together at their centromere. So this cohesin associates with chromatin at S phase only. It associates with chromatin at S phase. From that phase itself, these sister chromatids are held together with the help of cohesin. Okay, so during anaphase, what happens to this multi-protein complex? How this cohesin will leave sister chromatids to separate? So there is a separase. There is a separase. Okay, it is a protein. Okay, this separase will degrade. This separase will degrade cohesin. We know that cohesin is associated with chromatin at which stage? S stage itself. As separates degrades this cohesin, this will rupture, or this will it will divide cohesin from the chromatin. So before anaphase, so this is during anaphase. During anaphase, separase degrades cohesin. During anaphase, separase is a protein complex which will degrade cohesin. What happens before anaphase? So before anaphase, there is another protein called securin. Which keeps the separase in inactive state. So it keeps separase in inactive. I'll write it as inactive in bracket. So separase before anaphase, this is happening before anaphase. Before anaphase, separase is inactive. Therefore, it is not interfering with the separation. Okay. Therefore, it is not interrupting the cell cycle. Okay, the separase, as it is inactive, it is not degrading cohesin. Okay, so how it is inactive, how it is kept inactive? It is through this securin protein. So first, in order to activate separase, securin should leave this separase. So this destruction of securin. So if there is no securin, then separase becomes active. So now how to degrade or how to destruct this securin in order to release our separase? It is by APC bar C. Okay, MCDK activates. So how this APC bar C is activated? So we know now APC bar C will help in destruction of securin. How this APC bar C becomes activated? When MCDK activates this APC bar C by phosphorylating. When MCDK phosphorylates this APC bar C becomes what? Activated. This APC bar C once gets activated, functions in coordination with two homologous mitotic coactivators. No active APC bar C is not much it is not enough. Okay, so there are two coactivators along with APC bar C, that is CDC20 and CDH1. Now activated APC bar C along with the coactivators. So this initiates ubiquitination, and this leads to destruction, or you can say degradation of securin. So when MCDK phosphorylates or activates APC bar C along with the coactivators that is CDC20 and CDH1 will initiate the ubiquitination and degrades the securin. So once there is degrading of securin is done, once the destruction of securin is done, so then there is free separase. So what happens when there is free separase? It will destruct or it will degrade the cohesin. So what happens when cohesin is degraded? Once the cohesin is degraded, it will allow the sister chromatids to separate. That is what needed for anaphase. So after separation, so now it is like this. When this is the case, so what are activated, what are inactivated in this stage? So securin is bound to separase, making it inactive. How these are held together? Cohesin, which is now active. After this, what will be the situation? That is, what happens to this next? This becomes loosened. At this stage, what are all changes has been done? Securin is destructed. Separase is active. Cohesin leaves. Cohesin is degraded or destructed. So now this is known as sister chromatids. After separation it becomes chromatid. Now it is called chromosome. As it moves to separate cells, now it is called as chromosome. It is separated now in the metaphase plate. Then what next? After separation, it is pulled slowly towards opposite poles. Okay, all of the newly separated chromosomes move at the same speed, typically about 1 micrometer per minute. So this is the speed at which the chromosomes move. Chromosome in the sense this separated chromatids. Okay. So we can divide anaphase into two distinct stages, that is stage A and stage B, or we can say anaphase A and anaphase B, or we can say as early anaphase and late anaphase. Early anaphase and late anaphase. So the first referred to as anaphase A, it is the initial poleward movement of chromosomes. So it is the initial stage, it is the early stage of anaphase where initial poleward movement of chromosomes happens, which is accomplished by shortening of kinetochore microtubule. As kinetochore microtubule is pulling the separated chromosomes towards the pole, so kinetochore is obviously getting shortened. Okay, in this case the force is generated by microtubule by what? Depolymerization at the kinetochore. So depolymerization will lead to what? Will lead to loss of tubulin subunit at the plus end. So therefore the kinetochore microtubule is becoming short. So the movement towards the spindle pole is facilitated by kinesin 7 and kinesin 13 motor proteins that are localized at where? Kinetochore. So kinesin 13 is also there, but they do not have motor activity. So in this stage So there is initial poleward movement of chromosome, which is associated by shortening of KMT, that is kinetochore microtubule. This shortening is due to depolymerization at the kinetochore, which results in the loss of tubulin subunit at the plus end of the as the kinetochore moves towards the pole. So what about Anaphase B, that is late anaphase? What happens in this Anaphase B? So Anaphase B depends on motor proteins at the poles. Okay, here our interest will be at the pole. Okay, so it depends on motor proteins at the pole that pull the poles apart. So this pulls the poles apart, as well as on motor proteins at the central spindle that pushes the pole apart. So during Anaphase B, two poles move apart. Two poles move apart. Okay, it happens due to a sliding. So here it was shortening of KMT. Here it is because of sliding of polar microtubule past one another, and pulling forces exerted by astral microtubule. So this is directed by kinesin 5. The plus end directed kinesin 5 motor protein help in pushing of the which help in cross-link, adjacent, overlapping, antiparallel microtubule, help in sliding the microtubule past each other, thereby pushing the spindle poles apart. So in addition to this kinesin 5, there is dynein motor also that anchor astral microtubule plus ends to the cell cortex, pull the poles apart. So let us see the images one by one now. So at this stage, at anaphase stage, the kinetochore microtubules pull the two kinetochore towards the opposite poles. Sister chromatids do not separate because they are held together by multi-protein complex called as cohesin. So cohesin is associated with chromatin at the S phase itself. So during this phase, separase degrades the cohesin, so that will result in freeing of chromatin network or sister chromatids. Before anaphase, so there is a securin protein which keeps the separase in the inactive stage. See, anaphase begins, so the securin is destructed by APC bar C. Activated APC bar C will ubiquitinate and degrade the securin, leaving the separase free. That separase will degrade cohesin, which will lead to the separation of sister chromatids that are held together at the centromere. Anaphase after continuing, so there are two phases, that is anaphase A and anaphase B. Anaphase A is where it is the initial poleward movement of the chromosome. So that is anaphase A. Anaphase B is where the polar sliding movement of the polar microtubule push the poles apart. Anaphase A, this is anaphase A and this is anaphase B. And in anaphase A, the chromosomes are moving apart. In anaphase B, the poles are moving apart. See, MCDK activates APC bar C. APC bar C function in coordination with CDC20 and CDH1. It will degrade the securin, which will help the separase to be free, and separase will cleave the cohesin complex, and this cohesin complex was present in metaphase, which was holding the sister chromatids together. After the cleave or after the destruction of cohesin takes place, so the cohesin allows the sister chromatids to move apart. Next, chromosomes move by two independent and overlapping process. So that based on that, anaphase is divided into two distinct stages, anaphase A and anaphase B. Anaphase A, it is the initial poleward movement of the chromosomes, which is accompanied by shortening of kinetochore microtubule, that is especially the depolymerization at the kinetochore plus end, loss of the tubulin subunits here, which will help in shortening. That shortening will help the poleward movement. Okay, this movement is facilitated by kinesin 7 and kinesin 13. Next, what about anaphase B? It depends on motor proteins as well as motor proteins at the central spindle that pushes the poles apart. So this is movement of poles apart. So this is done with the help of polar microtubule, kinesin 5 motor protein and dyneins also. So that's all for today. In our next class, we'll be discussing about telophase. Thank you.